Hey, everybody. I hope that you're having a good day. This is a different plan than I had initially gone about, but this is how we're going to make a minor change. I'm planning on recording next Monday, or sorry, Tuesdays and Wednesdays lectures a little bit early as well, so that then you can have those and access them over the weekend. But also, that means that we can effectively move um, Tuesday and Wednesday's lectures to more or less be focused on office hours and answering general questions that you may have. But here, what we're going to do is we're going to get through our discussion and conclude our discussion of lipids, which will take us into a discussion of membranes as well. Now, with that said, we're going to go ahead and get cracking where we concluded our last lecture, and that was on the topic of triacylglycerols, also known as triglycerides. Again, I have those things on my screen that I will try to minimize so that you can see everything. But this is where we're going to get started. Interpreting triacylglycerols, also known as triglycerides. So one important molecule to have as kind of like a background or foundational molecule is this guy shown over here on the left, glycerol. Glycerol is 3 carbons and 1, 2, 3 hydroxide groups. Now, when you look at that molecule and you look at these hydroxyl groups, that gives it a little bit of polarity. So this molecule does have polarity to it. Now, when you have glycerol, you can esterify it. So you can add a fatty acyl group, so a fatty acid, what we've been talking about previously. And you form this, what's known as a triester. So 1, 2, 3 esters. So 3 esters have been added to a triacylglycerol to give you a triacylglycerol. So a fatty acid triester of glycerol is also known as a triacylglycerol or a triglyceride. Now, most of these triacylglycerols or triglycerides, which I'll try to use the term triglycerides more than not, triglycerides, contain 2 or 3 different fatty acids or types of fatty acids. So here, what we have is 1 palmitoleiol, 2 linaloyl, 3 stearoyl glycerol. So here's our glycerol backbone. On carbon number 1, you have a 16 carbon long monounsaturated fatty acid, and that happens to be palmitic acid. On carbon number 2, you have a linoleic acid, which is a polyunsaturated fatty acid. And then on carbon number 3, you have stearic acid, which is a fully saturated fatty acid. So if you think about, okay, well, what's our numbering scheme? What's our priority? Well, our priority is going to be giving our, if you think about it as, well, our numbering could have been 1, 2, 3, which is what it is, or it's 1, 2, 3. Given these 2 options, I'm going to label them A and B respectively. A makes our saturated fatty acid a lower priority, let me rephrase that, a lower numbered, therefore a higher priority than model A, where we have a monounsaturated fatty acid. So A is what we use because that gives our monounsaturated fatty acid or a unsaturated fatty acid the higher priority and therefore the lower number. So that's what you want to kind of seek out and look out for. Now, with respect to this name, I told you guys, and I stand by this, I'm not going to expect you to know the common names of fatty acids. There will be some that either through this class or in metabolism, you'll get a little bit more familiarity with, but I'm not going to expect you to know those names. Now, to go back to the physical properties of fats, animal fats have a high percentage of saturated fatty acid residues. Plant fats typically contain a higher percentage of unsaturated fatty acids, so more double bonds. Now, the melting temperature of a fatty acid varies with the degree of saturation and chain length. We've already talked about that. But this figure right here is comparing olive oil, which is a fat that is coming from a plant. Now, olive oil, what we see here is natural fats at 25 degrees Celsius. Olive oil, if you think about that at room temperature, go to your kitchen. If you have extra virgin olive oil or the EVOO, which I've seen on the internet, if you have any of that, you're going to notice that it is absolutely a liquid at room temperature at 25 degrees Celsius. Meanwhile, butter. Well, if you have butter and it's in your kitchen, and, in Central Texas at this time of the year, it's a little bit hard to keep your kitchen at 76 or 74 degrees Fahrenheit or 23 or 25 degrees Celsius. Chances are it's creeping up to about 28 degrees Celsius, in which case that butter is maybe not so solid as it once was. It's a soft solid. And in Central Texas, it's a very soft solid. What do we have there? Well, our percentage of saturated fatty acids is higher than that of our extra virgin olive oil. Our percentage of 16 and 18 carbon long unsaturated fatty acids is smaller than that of olive oil. Now, beef fat, the last of them, that's a hard solid at room temperature. If you think about ordering a steak, well, chances are if you get that steak, there might still be chunks of fat in it. And what's the temperature of that? Well, for starters, it's already been heated up a huge amount. So it's endured some heat. I apologize. Um, but at 25 degrees Celsius, if you get a steak out of your fridge and you leave it at room temperature because you want to throw it on the grill, well, that's going to be a solid. It's going to stay a solid. Even as it goes from 4 degrees Celsius to 25 degrees Celsius, it's going to be a solid. And what do you have a ton of there? You've got a considerably higher, nearly 50% of your fatty acids are saturated fatty acids. Now, what's the biological role of triacylglycerols? They function as energy reservoirs in animals. The fat content of a normal human allows them to survive starvation for 2 or 3 months. Triacylglycerols function, in addition to that, to insulate. Now, there's a couple of kind of anecdotes and stories that I always like to bring up. One of them is back in the mid 1990s, there were Wow potato chips. Now, Wow potato chips have been discontinued since I think the early 2000s, maybe it was the late 1990s. But these Wow chips were these chips that were believed to be low fat, and they were even considered to be like zero fat. And the reason for that is because they had a carbohydrate-based fat substitute. So what they had was they had a molecule that looked like this. So you got your six-membered ring, your pyranose, and then if you think about carbon number 6 on a glucose ring, you've got a hydroxyl group. Well, let's jump back a second. If you think about glycerol here, and then you compare that to glucose, a ring of glucose, what do they have in common? This, and this. They've got these hydroxyl groups. Those hydroxyl groups are great for the addition of fatty acids. So I bring that back up because there were food scientists that they took, I can't remember if it was glucose or what exactly, but they took a sugar. I'm going to go with glucose as my sugar. And they said, oh, that's got a bunch of hydroxide groups. Let's esterify them. And then let's throw some fats on there. And you might think, wow, well, Dr. Gray really can't draw a fatty acid chain very well. I concede you're right. But you also might be thinking, well, why would they do that? Why would you want to ingest more fat? And the truth is that fats have they give you kind of like a sensory satisfaction. Like you bite into a potato chip, like a baked Lay's versus a regular Lay's. They have a different texture to them. Something that's been deep fried in vegetable oil is going to taste and feel a little bit different. Now, what these researchers did was they made this pyranose structure. They threw a bunch of fatty acid chains on there. Here we go. I'm just going to get lazy on these. So that whenever you bit into these chips, they tasted and they had that same sense of being like a regular potato chip with a full fat chip. So you had these chips that you'd bite into and it tasted like and it felt like a regular chip. But the difference is this olestra, this carbohydrate based fat substitute, you didn't metabolize the same way. You had all those fats, and so it kind of scratched part of that itch, but there's something that your body was like, oh, this is different. Now, this is really popular. This is a really good strategy because you wouldn't have that caloric load of a regular triacylglycerol. But the problem is that whenever you would eat these chips with Olestra, lots of people had really negative gastric experiences with it, to put it bluntly, or to put it lightly, not bluntly. And so it didn't take long for that to kind of be a reputation and a problem that even the best marketing team could not rebrand and spin to be something that people were compliant and accept and willing to accept. Okay, so that's one thing. Another thing is I always like to bring up whenever I look at this fat content of normal humans allows them to survive starvation for 2 or 3 months. There's this guy who his last name is Barbieri, and I've forgotten his first name at the moment, but he was in a position in like the early 1970s. Um, he was considered to be like dangerously morbidly obese, and he fasted for I think it was like 375 days. And in that time, he lost like 240 pounds. He drank nothing but water, took a multivitamin, and really did not ingest any sort of calories or anything of that nature. And lo and behold, he survived. Well, why did he survive? Because he had lots of fat stores in his body. So I just want to, I always like to bring that up to stress how much energy fats and triacylglycerols and fatty acids can provide you with. Now, when it comes to fats and their and how they're metabolized, there's actually if we look up top here is a triacylglycerol. There's two major ways that I think is important to understand their processing. One is through an industrial process. Now, if you ever read the book or seen the movie Fight Club, you know you're familiar with this. This process is known as saponification, where basically you take fats or a fat source and you throw them in a strong base solution like sodium hydroxide. And this is called rendering. And basically, you use this to make soap. Now, that soap is fats or sodium salts of fatty acids. So, your a bar of Dove soap is really just basically a bunch of fats. Now, I mentioned this in the context of Fight Club because what they did in that movie and in that book was they took um, different fat sources and they rendered it to make soap. Now, that's an industrial process. So, soaps that you use, that's the process that's involved. Now, what does your body do by comparison? Well, your body uses enzymes. Not too much of a shocker there, but it hydrolyzes them. So it uses types of enzymes known as lipases. And a lipase is going to release a glycerol molecule as well as an ionized fatty acid. So it's going to take a triacylglycerol and split it into glycerol and three fatty acids. Now, when we look at a triacylglycerol, ultimately, if you remember, I was showing you that glycerol has like a little bit of polarity to it. And that's like, oh yeah, that's cool because those three hydroxyl groups. But when you esterify it, okay, this is where our hydroxyl group was. And it gives you that carboxyl group. And oh, that's there's a partial negative, partial negatives. But let's think about a fatty acid. That R sub one, that R group, what is that? Hydrocarbons. That's a long alkyl chain. It's just a bunch of carbons and hydrogens. So this would be considered a non-polar lipid. So despite glycerol being semi-polar, a triacylglycerol is a non-polar molecule. Now, a glycerophospholipid, by comparison, let's take a look at that. A glycerophospholipid, glycero, okay, we've got that glycerol backbone. Okay, there's our 3 carbons of glycerol. It's been esterified. There's 1, 2 fatty acids. Okay, but another part of this glycerol molecule, it's been phosphorylated. We get a phosphate group added to there. Then last but not least, on that phosphate group, we get something else added to it. This is known as a head group. And this head group, this X molecule right here, and this phosphate group make glycerophospholipids polar lipids. Now, our last group over here, sphingolipids. I gotta be honest, sphingolipid for the longest time was my favorite science word. And a sphingolipid is built on an 18 carbon long backbone. The difference is, and I always like this depiction of it because these 3 carbons right here, they look similar to your glycerol. Now, the difference between them is that, well, it's not glycerol. It's not the same thing because if you look, yeah, there's a hydroxide group, but then there's this alkyl chain at the end of that, which gets us to that 18 carbon long chain. Now, a sphingolipid is going to have some variations on it. One thing that it's going to have is it's going to have this amine on this carbon right here. Then in place of a hydroxide group on this carbon, we're going to have a phosphate and a head group, much like what we see on a glycerophospholipid. Now, your nonpolar lipids are going to be your energy stores, so your triacylglycerols. Your polar lipids are the basis for your bilayers. So when you think about a lipid bilayer, and if you've ever taken a cell biology or molecular biology class, when somebody draws a lipid bilayer, they draw it like this. 2 tails. 2 tails on the end of that head group. The reason for that is because this is your polar head group, polar, I label it HG, and then these are your nonpolar tails. So these are our nonpolar tails. Okay, so we got a polar head group and nonpolar tails. Now, there's only 2 of those tails, which we see with our glycerophospholipid here and here, respectively. Now, glycerophospholipids. Glycerophospholipids, to kind of focus in on those, they are the major lipid component of biological membranes. They're amphiphilic molecules with nonpolar aliphatic tails and polar phosphoryl head groups. This word right here, amphiphilic, that is a word that is simply used to define a molecule that has a polar region and a nonpolar region. Glycerophospholipid is a great example of that. It's got a polar head group and a nonpolar aliphatic tail, or sorry, nonpolar aliphatic tails. Now, this X right here, I want to focus in on that. Like I said, this is the polar head group. Those polar head groups can be any one of these different molecules. So that X that I just showed you that I'm circling now on this repeated glycerophospholipid could be any one of these molecules. Now, if we look at something like phosphatidylglycerol, what I am saying is that this whole thing can take the place of that X. When that whole thing takes the place of that X, oh man, aliphatic tail, aliphatic tail. Yes, I am saying that a polar head group can have another glycerophospholipid on it. These become very complex. This is known as diphosphatidylglycerol or cardiolipin. These molecules, what they are involved in is, think about these as being a part of a membrane. These are involved in signaling. These are involved in communication. These are involved with one cell telling another cell what's going on. The presence of these different molecules sends different messages. Think about this almost like a yard sign. Like this, if there's a bunch of phosphatidylglycerol displayed on the exterior of a cell, that's like saying, hey, this is what's going on inside this cell. Some of these molecules are involved in signaling like a cell is under distress. They would be something like a pathogen has been detected, the cell is undergoing senescence or autophagy or programmed cell death. And that's what's going on in here. So that pathogens may be coming for you. So get ready. Now, do I want you to know any of these specific head groups? No, I don't. What I want you to know about them is how to basically visualize them. So if we look at serine, for example, this right here ends up right there. Now think about the amino acid serine. I always draw serine like this, NCC. H3 plus. Then we've got CH, because this right here, I'm underlining your alpha carbon, CH2, and now I'm putting a dot under that carbon, and that is this carbon. Because again, with serine, we've got an OH there. Well, what has replaced that OH? Well, what's replaced that OH is this whole thing. Okay. So I want you to be able to translate basically one of these structures into, translate from this, from the structures that you know, to something like this. All right. Now, sphingolipids. Sphingolipids are major membrane components. Now, they are derivatives of an 18 carbon long amino alcohol known as sphingosine. The basic unit of a sphingosine is a ceramide. Okay, so on the far left here, we've got a sphingosine. Here's our 18 carbons that I alluded to earlier. And then here is our amine on this carbon. Okay, so this is an amino alcohol. Now, in order to make a sphingolipid, we take sphingosine and we take this amine right here, and that is where we add a fatty acid residue. Now, this shown over here on the right, this is an example of a completed sphingolipid. What we have is a sphingomyelin, a palmitic or a palmitate residue right there on this amine. Now, sphingomyelins, the head group for a sphingomyelin is phosphocholine or phosphoethanolamine. So this group right here, depending on what is there, it's either going to be phosphocholine or phosphoethanolamine to make a sphingomyelin. Now, cerebrosides are examples of sphingolipids, in which case their head group would be a single sugar like glucose, as is the case with a glucocerebroside. So this right here would be replaced with, I'm going to draw my, no, that's not what I'm looking for. There we go. Down, up, down. Nope. Something like that. So that would be an example of a cerebroside. Now, I think that for you guys, it's going to be important for you to recognize and know what these are derived from, as well as the common head groups for sphingomyelins and cerebrosides, respectively. Now, these molecules get processed. Glycerophospholipids, specifically, they're going to be processed in a unique way. And depending on the enzyme that is involved in processing them, you're going to get a different product. So if we have phospholipase A1, phospholipase C, and phospholipase A2, and phospholipase D, these are cutting at different positions. So if you look at phospholipase A1, that's going to cut right here. It's going to release this group. So it's going to release a fatty acid. Now, if you look at phospholipase A2, it's also going to release this, a fatty acid. Phospholipase A1 and A2 effectively cut in the exact same location on the fatty acid side of this oxygen, as opposed to cutting on the glycerol side. Now, phospholipase C and phospholipase D, these are going to cut in different locations, and that C is going to cut to release a phosphorylated head group, whereas D will cut to leave the phosphate behind and release only the head group. So that X, remember, could be any one of these molecules right here. So phospholipase C and D, you're either releasing the phosphorylated head group or the head group alone. Phospholipase A1 or A2, you're releasing the fatty acid alone. Now, cells are going to continuously degrade and resynthesize membranes and sphingolipids. So these things are under constant turnover, being replaced, replenished in order to maintain like a membrane with high integrity. Now, I do want you to know where these different lipases cut and what their products are. What I would say is to think A1 and A2. They basically cut in the same location, but which carbon number is it? C and D. Well, C, you're getting I'm going to say phos head group. And D is just getting head group. A1 and A2, those two are going to release fatty acids. Okay, now, in addition to our, well, heck, if we go back, we talked about fatty acids. We did that the other day. We talked about triacylglycerols and triglycerides today. We talked about glycerophospholipids and sphingolipids, two major parts of membranes. Now we need to look at lipids and their non-membrane functions. There are signaling lipids. For example, eicosanoids, they mediate inflammation and they give fever and pain responses, or they mediate or initiate that. Steroid hormones act as chemical messengers. There are also lipid cofactors, ubiquinone. I know some people like to say ubiquinone. This functions as an electron carrier in the electron transport chain, or ETC. There are some other lipid cofactors that do effectively the same thing in photosynthesis. They're just carrying electrons. Now, I mentioned this the other day. Hearing about oxidation reduction, not exactly the most exciting thing on Earth. I mean, it's in the top 30, but it can feel a little bit tired. It can feel a little bit dry. Photosynthesis and the electron transport chain. Those are two processes involved in cellular metabolism of fuels. You're extracting energy. The movement of electrons transfers a huge amount of energy. So these lipid cofactors play a big critical role in that. One last non-membrane function of a lipid is pigments. They serve as pigments and antioxidants. Vitamin A is an essential pigment in vision. Vitamin K functions as an antioxidant. And then vitamin D, which is also a lipid. Vitamin D is a fat-soluble vitamin, is involved in overall liver health, bone health, and it's one that has been implicated or was implicated long, long ago in helping people avoid enduring the rickets. Okay, eicosanoids. At a very low concentration, these are involved in the production of pain and fever, the regulation of blood pressure, blood coagulation, and production and reproduction. These are produced and used locally. Now, what does an eicosanoid look like, and how is it released? Okay, so here is a membrane phospholipid. Now, this is going to be a glycerophospholipid. Phospholipase A2 takes this molecule. This is 20 carbons. I believe eikos is 20 in Greek. Now, this 20 carbon long molecule is going to once it's released from a glycerophospholipid, it's going to fold into this molecule right here, known as arachidonic acid. Arachidonic acid, whenever that is released from a glycerophospholipid within a membrane, it can go on to form this molecule right here, known as prostaglandin E1, or it can form leukotriene, a leukotriene, or a thromboxane. Now, what these things are going to do is, or the way that this is going to be done, is with enzymes known as COX enzymes. Cyclooxygenases is what they're known as. what they're going to do is they're going to take this 20 carbon long structure and cyclize a portion of it. So they're going to make a little ring structure there. And whenever they do that, and whenever prostaglandin E1 is formed, well, that's going to initiate, like you're gonna feel that. You're not gonna feel particularly good. You're gonna have, either you're gonna have a fever, or it's something, you're going to be in pain, or your blood pressure is going to be elevated. You're going to experience some kind of negative side effects of this. Now, what this is showing right here is that NSAIDs, or non-steroid anti-inflammatory drugs, block the formation of these prostaglandin thromboxane. So they block this cyclization of arachidonic acid. So they prevent the formation of this, and thereby they prevent that fever from becoming an issue. These are known as COX inhibitors. So your non-steroid anti-inflammatory drugs are COX inhibitors. They block that cyclization. Examples of this are Singulair and Zyflo. Okay, now we have, let's see, how many more? Yeah, about, not too bad. Not too many more slides in this slide deck, but we proceed. So cholesterol is based on a core structure consisting of 3 6-member rings and 1 5-member ring, all fused together. That's what it looks like. That is cholesterol. Cholesterol is a steroid. It's the most common steroid in animals and a precursor for all other steroids in animals. There are molecules known as phytosterols that people can take. These are plant sterols. They come from plants, and they're found in plant membranes, and they help maintain membrane fluidity. They control cholesterol serum levels whenever you take them as a person, or whenever a person takes them. A couple of other steroid hormones seen over here on the right, well, these are derivatives of cholesterol. Steroid hormones bind to very specific receptor proteins in the nucleus, regulating gene expression and metabolism. Major groups are the sex hormones, and these are produced, and those produced by the adrenal cortex. For example, testosterone and estradiol. Now, lipid-soluble vitamins. A, D, E, and K are your common ones. A and D, these two vitamins were discovered by the same people. They worked in the ag department at the University of Wisconsin, and what they studied was basically, well, how can we find an ideal diet for cattle? And the fascinating thing about the people who discovered these, was 1900s or so, was that they realized that it was pretty much cost prohibitive to do these studies on 500 heads of cattle, because, cows, well, they might live for 5 years. And so what they did, I don't know how long a cow lives, but I'll say it lives longer than most fish. What they did was they actually studied rats. So what they did was they found and bred rats, and they would keep them in laboratory conditions, and they would give them specific diets. And what they were able to do was they were able to find out, oh yeah, cod liver oil was something that they needed to give to the rats to make them healthiest. And as it turns out, cod liver oil is very rich in vitamin D. Now, vitamin D regulates calcium and phosphorus metabolism, and so this was a benefit to them. Vitamin D is also very helpful for humans in that it prevents rickets, RICKETTS. I don't think there's an E at the end of that. Yeah, it prevents rickets, which is something that causes your bones. It's usually in it's very young children. Their bones kind of bow out, their legs kind of bow out. Now, vitamin D is actually a group of structurally related compounds that play a role in the regulation of calcium and phosphorus metabolism. The most abundant form in the circulatory system is vitamin D3. Now, one of the things that people will regularly say about vitamin D, which is indeed true, is that, oh, go out in the sun, you'll get your vitamin D. And it's true that it's true, and it's also kind of a misrepresentation. Maybe not misrepresentation, but what is necessary for vitamin D is you have to have UV light exposure, which is complicated in the sense that too much UV light exposure can be damaging. It can cause skin cancer, whereas just the right amount of UV light exposure, and boom, you get the type of vitamin D that you need, and that is this cholecalciferol, this vitamin D3. Now, after that UV light exposure, your liver function must be intact. And then after your liver, your kidneys must function properly. So UV light followed by liver, followed by the kidney, and boom, you've got this 125 dihydrocholecalciferol, where these hydroxide groups have been added by the um, liver and the kidneys respectively. I can't remember which one. I think the first one, this position at position one, that's added by the liver, and then at position 25 is added by the kidney. I can't remember exactly. It's not completely important for this. But this metabolism, you have to have a healthy liver, you have to have a healthy kidney in order for you to have the right vitamin D that you need. Now, if you take a vitamin D supplement, if I remember correctly, you are taking this stuff right here, this 125 dihydroxycholecalciferol, or dihydroxy vitamin D3. Vitamin E, also a fat soluble vitamin, and it's a group of compounds of similar structure. The most active form is alpha tocopherol. It's an antioxidant. It traps free radicals, and these, which are formed through the result of the oxidation of O2 of unsaturated hydrocarbon chains and membrane phospholipids. This long chain right here is made up of molecules known as isoprene units. These isoprene units are essential for the synthesis of cholesterol. And these are great for carrying electrons. So this long chain right here is really great at basically bouncing electrons from one position to another position on there. Okay, so that was our summary, our everything that we need to know about lipids. Now what we're going to do is we're not changing gears completely, but we're going to hit some high points on membranes. So first and foremost, what does a membrane do? Well, a membrane serves as a barrier to a toxic chemical. It keeps, in summary, it keeps the bad stuff out and the good stuff in. It can facilitate cellular motion. It can assist in reproduction. It can modulate cell signal transduction and cell-to-cell interactions. Apologize. Now, there's three structures that I want you to be familiar with. If you look at this one in the lower right-hand corner, this is known as a micelle. This is something I remember seeing for the first time when I was in organic, I believe organic 2 was whenever we saw it. And I want you to look at a micelle. Well, its shape, spherical. Now, let's look at this. Individual units are wedge-shaped. Okay, cross-section of head greater than that of the side chain. Okay, so we've been looking at these head groups and imagining that they're polar. And if those are polar, then what is on the other side of it? Well, it's non-polar. Okay, so if you look at these non-polar groups, and then you look at the exact center there, what can you tell me about the polarity of a micelle? Well, this interior right here, that's a non-polar region. So the interior of a micelle is non-polar, the exterior is polar. Okay, another thing that you can tell me about the interior of a micelle, well, there's only one aliphatic chain. Interesting. That helps them be a little bit smaller. Now, if we transition and go over to a bilayer, hearing the phrase lipid bilayer isn't too crazy of an idea. We've got that polar head group and then non-polar tails. Again, we've got two non-polar tails, and the individual units of these are cylindrical. Okay. And last but not least, if we compare that to a liposome, Okay. Shape, spherical. Each one of these individual units, well, it's spherical. So a liposome, it's got that is your non-polar region. And then on the inside, oh, that interior, that's a bunch of polar head groups. The interior of a liposome is polar. That's an aqueous environment in there. Compared to the micelle, the interior of a micelle is nonpolar. Now, that's significant because you could kind of think about a liposome, and the way that I think about a liposome is it's basically like a cell membrane scaled down. That's a little bit too simplistic of a view, but I think it's always helpful for me to visualize applying a bilayer to a liposome and saying, okay, well, if I'm thinking about this as the plasma membrane, it's just going to blow up a whole lot and there's more complex things in there. Now, movement of lipids or glycerophospholipids or sphingolipids within a lipid bilayer. If we're going from this side to this side, we'll refer to these sides as leaflets. They are known as the outer and inner. Imagining that in here where I'm drawing that's inside cell, in cell. To go from the outer to the inner, that is known as transverse diffusion. And we have this flipping that's taking place. Now, if we're going from staying in the outer leaflet and we're just kind of moving from one position to another, I always kind of visualize this as being like if you go to a concert, and you're like, oh, I want to get close to the stage. Can you do that? Yeah, just you bump into people, you see an opening, you step in there, you go, and then boom, you're at the front. This is known as lateral diffusion. This happens very rapidly. Now, one of the reasons that happens rapidly is because a lipid bilayer has two different major states, a paracrystalline state and a fluid state. And in order to transition between these two different states, there's what's known as a transition temperature. Whenever the temperature is below the transition temperature, the lipid bilayer becomes more rigid. It basically becomes a gel-like solid. When the temperature is above the transition temperature, the bilayer is known as a liquid crystal. Okay, so that would be this down here. Everything's moving. It's chaotic. That's why I like the concert analogy because yeah, it's chaos. You know, the concert starts and there's general admission. You're just moving all over the place. If you're standing with a friend, you hold hands because otherwise one person's going to the other side of the show. The transition temperature, however, is dependent on chain length. Okay, let's pause and think about that. Chain length and temperature. Have we thought about that before? We have. We thought about that when we were talking about individual fatty acids. The longer the fatty acid chain, the higher the melting point. If you put a double bond in there, then your melting point goes down. But we have this connection between a physical property and a trait of that molecule. So the transition temperature is dependent on chain length and the degree of saturation of its fatty acid component residues. Now, cholesterol is going to broaden the temperature range of the phase transition. Now, whenever I think about cholesterol, I always think about like, well, you know, dietarily, some people are on low cholesterol diets. You can't have too much because if you have too much, then what happens? Your arteries begin to harden up. So cholesterol, one thing that it does is it impacts the fluidity of a lipid bilayer. Now, I love this experiment and I love this figure because it gives you that point. So to set the stage a little bit, this is from a paper that was written in the 1960s. So in 1962, there were researchers, Marr and Ingram, who studied the effect of temperature on the composition of fatty acids in E. coli, or Escherichia coli. I always like to say Escherichia coli whenever I have the chance. So they studied the effect of temperature on fatty acids. What they did was they grew E. coli at 10 degrees, 20 degrees, 30, and 40 degrees. Now I'll tell you, E. coli's ideal growth temperature is right about here, 37 degrees. And what I mean by that is it will grow its fastest, and basically every cell will live from generation to generation. If you heat it up too much, what's going to happen? Well, then E. coli is going to die. So and whenever I say heat it up, I mean if you go to like 50 degrees, most of that E. coli, I'll say more than half of it's probably going to die. Now, if you go to a lower temperature, below its ideal temperature, well, it will live, but it's just going to do it very slowly. Um, so when we grow at 10 degrees Celsius, when we grow E. coli, I'm going to scribble all this out because all that we're focused on, and all that I want you to focus on, is this first column. When you grow E. coli at 10 degrees Celsius, what happens? Well, the answer is it grows. And then the second point is, well, we've got fatty acids. We look at myristic acid, palmitic acid, palmitoleic acid, and oleic acid. Palmitoleic acid and oleic acid are both monounsaturated fatty acids. So if you compare a monounsaturated fatty acid to a fatty acid with the same number of carbons, but it's fully saturated, the melting point of these monounsaturated fatty acids is going to be lower. So that means it's more than likely going to be a liquid at room temperature. So it's more liquidy. Now here what we have is what is the percentage of the total fatty acids in those cells of palmitoleic acid and oleic acid. And that'll be 38 plus 26, that's 24, 64% of the fatty acids when E. coli is grown at 10 degrees Celsius, 64% is made up of saturated or monounsaturated fatty acids compared to 22% that are saturated fatty acids. Now, that just shows that, well, at a lower temperature, we have more monounsaturated, which I just said was Mu to saturated. So more Mu to S. Now, let's jump over to 40 degrees, close to the ideal temperature. And let's look at our monounsaturated fatty acids. At 40 degrees Celsius, close to the ideal growth temperature, only 21% of the fatty acids are monounsaturated. So we see that drop from 64 to 21. Okay, so the abundance of the monounsaturated fatty acids got cut 2/3 basically. Now, if we look at the saturated fatty acids, because maybe it's that the monounsaturated, maybe there's more polyunsaturated fatty acids. I don't know. Well, when we look at the saturated fatty acids, we have about 56%. So we've gone from 22% at a low temperature to 56%. What does that say? 22 to 56, that's multiply it nearly by 3. Well, let's think about those saturated fatty acids. Our melting point is higher. So let's switch this and let's not talk about them as saturated or unsaturated. Instead, let's say, are they going to be a solid or liquid at that temperature approximately? So these first two, are they more than likely going to be a solid or liquid at 10 degrees Celsius? Oh, they're almost absolutely going to be a solid. What about these other two? Well, at 10 degrees Celsius, they're going to be a liquid. So I think about this kind of like, this was 64 to 22. We've got about I always kind of think about it as like almost votes. We've got 3 votes for liquid and 1 vote for solid. So you could kind of think about this as our membrane is like 75% liquid at 10 degrees Celsius. Now, then when we go to a higher temperature, we've got 56% solid and 21% liquid. Okay, wait. But we've changed. We are closer to our melting point. So, yeah, it's a solid at room temperature, but are we at room temperature? No. So, although this is more solid-ish, well, we're closer to our melting point. One of the ways that I think about these 2 columns and across all 4 of these columns is I like to imagine it as these E. coli cells, whether they're at 10 degrees, 20, 30, or 40 degrees Celsius, they want their membrane to feel the exact same way. Okay. At 10 degrees Celsius, they want it to have this much mobility, they want it to have this much bounce. And in order to attain that, they've got to have 64% of the fatty acids be monounsaturated, 22% be saturated. Okay, now, when our temperature is higher, we want to have that same level of fluidity. Can that be attained? Yes. How do we attain that? We attain that by using fewer monounsaturated fatty acids and more saturated fatty acids. So that's the biggest thing about this difference. Okay, now what I want to stress with all of this is that fatty acids influence membrane integrity. The physical properties of those fatty acids influences membrane integrity. Now, when it comes to a membrane, you have an inner and an outer leaflet. And you have something known as lipid asymmetry. This is really stressing the point that lipids and what's on the inner and the outer leaflet of a lipid bilayer, it's very dynamic. So typically, 50% of the phospholipids are going to be found on the inner leaflet, 50% of them on the outer leaflet. There are some lipids that they have a, 2-to-1 ratio of those that are on the outer leaflet compared to those that are on the inner leaflet. Then there are other lipids that, under normal circumstances, they are 100% on one side. Lipid asymmetry is, you've got a mixture. Some are going to be on the inside, some are going to be on the outside. And depending on the conditions and the circumstances, you might put a bunch of lipids on this side, but not on the other. Now, in order to adjust that lipid asymmetry, phospholipids are synthesized on or near the interior of the membrane. There are 2 mechanisms to get the phospholipids from one side of the membrane to the other. We kind of alluded to this, but now we're seeing the parts. Flipases, I kid you not, that is what the name of the enzymes. They are enzymes that catalyze the flip-flops of specific phospholipids in what is known as facilitated diffusion. So they'll take a lipid from one side of the membrane and move it to the other, and they will do it very rapidly. This is known as transverse diffusion. Now, in the absence of an enzyme, which is what we see right here, that's going to happen very, very slowly. Because think about this polar head group right here that I just put next to it. It has to move through this non-polar region. It's not going to like to do that. Flipases tend to equilibrate the distribution of the corresponding phospholipids across a bilayer. Phospholipid translocases transport specific phospholipids across the membrane in a process that is driven by the hydrolysis of ATP. Translocases can establish a non-equilibrium distribution of the phospholipid. Your membrane composition. Now, we don't have too many more slides left, but This is going to, kind of be on the downhill movement. Biological membranes contain proteins and lipids. Proteins catalyze reaction, mediate the flux of nutrient and waste, and participate in relaying external information. So they communicate, hey, what's going on outside? What cells are nearby? Okay, I'll let everything on the inside know. Now, protein-lipid ratios vary with membrane function. In addition to that, if we look at different organisms, E. coli, for instance, their components by weight, sorry about that, 75% of a E. coli membrane, its weight is wrapped up in proteins, 25% phospholipids, and lo and behold, 0% in cholesterol and sterols, because they don't have cholesterol. If we look at yeast by comparison, about 52%, more than half of its weight is wrapped up in proteins and phospholipids, and 4% in sterols. Now, if we look at a human myelin sheath cell, what do we have? About 30% is protein, 30% is phospholipid, and about 90% is sterols. These are kind of trivial pieces of information, but it's good to know if you look at these different organisms and understand, like, okay, some have no sterols and others, well, they use almost half of their weight for this. Now, membrane structure and assembly. The phospholipid bilayer is known as a fluid matrix. The bilayer is a two-dimensional solvent. Okay, here it is. This is everything. Now I've got another representation of this shown on the next slide with just a little bit more emphasis on, well, what are the proteins? I'm going to show you this one here, and then I'm going to go into more detail on the previous slide. And then we'll get into some of the nuts and bolts of those different things. So first, what I want to do is draw your attention to these two indicators. Are we on the inside or the outside? What is shown on the outside side, so above this, that is by design. Glycolipids, for example. A glycolipid is a branch sugar molecule coming off of a lipid. So this is a sugar or a glycinated lipid or a sugar-aided lipid. This is used for communication. There are some proteins that they have regions that are on the outside of the cell. Then there are other proteins, these right here, known as glycoproteins. These other proteins are, well, you got a polypeptide chain, but then you got all these sugars branching off of them. These are used for communication, and not just one cell telling another one, hey, there's there's a pathogen here. Sorry about that. But instead, this is like for cellular development, for the cell cycle. Like, hey, now that this glycoprotein is emerging on the exterior cell, this particular organ needs to go on to do X, Y, and Z. Now, on the inside side of a cell, okay, we've got proteins. Let's track this protein. Here's our protein right here. This kind of like elongated hot dog shape has a connection where it has this association with a phospholipid head group. Neat. Another protein, an integral protein. This has a transmembrane helix that spans the membrane. So this is almost like a protein that's been stabbed through the lipid bilayer. We have a peripheral protein that has been covalently linked to a lipid. So there's a covalent bond between this head group and this protein. Then we've got an integral membrane protein with multiple transmembrane helices. Okay, so proteins can be covalently linked to a membrane. They can be inserted with multiple alpha helices. And then we could also have these kind of interactions between head groups that potentially are just, well, ionic interactions between them. Now, these little hot dog shaped things right here, 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 those are steroids, lots of cholesterol. That's going to impact the fluidity of this membrane. The things that are drawn on this that are on the outside of the cell, they are there for a reason. The things that are on the inside, they are there for a reason. Now, this glycoprotein that I talked about earlier, oligosaccharides, that would be an example of a glycoprotein. Or an oligosaccharide is a sugar that would be a part of a glycoprotein. Now, integral proteins are associated tightly, and in order to extract those integral proteins from the membrane, you got to use a detergent. What do detergents do? They bust up hydrophobic interactions. Or you could use a chaotropic agent like urea or guanidinium hydrochloride in order to separate from the membrane. Lipid-linked proteins tend to be globular after they are released. And then peripheral proteins are globular-like, and they are easily dissociated. So this peripheral protein down here is kind of like this one right here. It's kind of like, yeah, it's there, but it doesn't need to be. And if it gets released, it gets released. Okay. This depiction, and I want you to know this, these models right here are different transmembrane proteins. I want you to know basically 7 pieces of information from this. No, 8. Yeah, 7 or 8 pieces of information from this. One of them, first and foremost, is looking here. That's an alpha helix. You guys know that. So that's not one of the pieces because you already know that. What I want you to know, and I want you to think about, is the amino acids within that region of that protein. And as a reference, we got one right here. But before you even look to that reference, think about it as, okay, well, what do we know about this region of a lipid bilayer? What I know is that it's super nonpolar. It's got those long aliphatic chains. Okay. So if this is nonpolar, what amino acids would I expect to see within this region? And now look at your reference point over here. We've got, and it's not 100%, but we've got a lot of important residues. ILE, isoleucine, LEU, leucine. Isoleucine, leucine, leucine, isoleucine, glycine, valine, those are nonpolar residues. So within that region of the protein, there's lots of nonpolar amino acids. Now, the next thing that I want you to know is I want you to know all these. I want you to know that a type 1 transmembrane protein has the N termini outside of the cell and the C termini inside of the cell. What about a type 2 transmembrane protein? Well, the N termini is on the inside and the C termini is on the outside. Type 3. Oh, well, the type 3, you've got multiple transmembrane protein regions, but this is one polypeptide. Type 4, we got 1, 2, 3, 4, 5. You got multiple transmembrane domains, but multi polypeptides. The type 5 is a lipid-linked protein where some part of the protein has been modified to have a lipid link to it. A type 5, well, it's got a membrane, it's got a membrane region, but it's also got a lipid linkage. This was type 5. The type 6, has a part of it is inserted into the membrane, but then it's also got a lipid linkage. Now, those are the things that I want you to know from this slide. What is the makeup of the amino acids within a transmembrane protein? Is there, I like to ask the question, which amino acid is most likely to be found within the transmembrane domain that goes through the membrane? And I'll give you aspartic acid or glycine or valine or whatever. And your answer would be, well, valine. Let's say I gave you serine, aspartic acid, and valine. Your answer is going to be valine. Why valine? It's a nonpolar residue. Okay, now, not all transmembrane proteins are alpha helices. Instead, there are beta sheets that can form, making what's known as a beta barrel. And they're going to give you something that looks like this right here. Where here, if you look on this side right here, those are a bunch of beta strands going in opposite directions. And they basically form this opening. Now, hemolysin toxin, or the alpha hemolysin toxin protein, what that does is it basically forms a hole, and basically lets nutrients and sodium ions bleed out from the cell. And whenever that happens, well, that cell's going to die. Why is it? Because because the balance of ions inside and outside is going to be distorted, and that cell is going to die as a result of that. Now, proteins can be lipid linked. I mentioned this a moment ago. How can they be lipid linked? Well, one way in which they can be lipid linked is, this takes a little bit to wrap your mind around, but here's our protein. Here's our C termini. And we get this isopeptide bond right here. So we get a protein that has this peptide bond connected to a phosphohead group, specifically phosphoethanolamine. We got a bunch of sugars that then connect it to phosphatidylinositol. And then this right here gets embedded to the membrane. That phosphatidylinositol, you might think, well, that looks familiar. This is a glycerophospholipid, or sometimes people know these as GPIs. Other proteins can receive lipids like farnesyl, this is an isoprene unit, or geranyl, which is just a long hydrocarbon chain. Now, there are 4 types of lipid-linked proteins. They can have a palmitic group. And what I want you to know here is I want you to know what amino acids can these different groups be added to. So palmitic group can be added to a cysteine or a serine. This is a thioester-linked fatty acyl anchor. You can have an N-meristic group added on the amino termini of glycine, an amide-linked meristal anchor. Then you could also have a thiol ether-linked prenyl anchor. So this would be a farnesyl group added to a cysteine. So your targets are going to be cysteine, glycine, or a cysteine or a serine. GPI linkages, well, that's what we just saw right here, but from a different depiction. So I'm going to draw an X for your protein, and then I'm going to draw another X for your protein. So GPI-linked proteins are located on the exterior surface of the plasma membrane and always are attached to the C-terminal residue. So to answer this, they're always attached to the C-termini as opposed to individual amino acids. Now, membranes have a tendency to roll across a submembranous region of proteins known as the membrane skeleton. These are ways to move things, and what they do is they use a bunch of different proteins to do this. Spectrin accounts for 75% of the skeleton and is composed of repeating segments that fold into triple-stranded helices. Now, what these are able to do is they're able to reposition different proteins that are able to move things. Membrane wraps. What I want you to know about membrane wraps is that they are regions of a membrane that are enriched in sphingolipids and cholesterol. What they're able to do is cluster a bunch of proteins together in order to carry out specific functions. They're rich in sphingolipids and cholesterol, but not in phospholipids. A number of transport proteins are associated with lipid rafts. For instance, your proteins that are involved in transporting ions from one side of the membrane to another. Okay, there's a lot of information, but that is the end of the material on exam number 3. I hope that was helpful. And like I said, I'm going to get to the other things hopefully over the course of the weekend. But in the meantime, have a good one. And if you've got any questions, please don't hesitate to reach out to me. We can set up an office hours over the weekend or something. Just let me know. All right, have a good